Hi, Mary Axholm here. So I'm trying to finish up Lab 2A, and this is the Bacterial Enumeration, Dilutions, and Plate Count lab experiment. And so the plates have been incubated for over 48 hours at this point. I flipped them at 24 hours as directed, and so the agar plate is upside down as directed. I'm going to show these each in the video here, as well as I've already taken pictures of them so that I can complete the lab. I'm turning this lab in late, and I'm actually really upset about it because it's such an intense lab, and at this point I think my grade's going to be pretty low. And I also realized that we needed all of what's in most of this lab to conduct the other labs. So anyway, it's almost done. So this is 10-1. I'm not sure how the lighting is in here, but as you can see, there is some growth in there. It's very hard for me to see. I noticed on the lab exercise, it said there's an example of 10 times, 1 time, and I think it was 100 times, but I don't see us being instructed to look at this under the microscope. But to me, this looks like well over 300 CFUs. So this one is pretty big. However, there are bigger, I don't know what these bigger dots are. See right there that I'm pointing at? That's a bigger dot. So there's only a few of those, and those are in most of each of these. So I'm wondering if that's something totally different. Could be. Maybe it was something that got into it. I don't know. But anyway, this is 10-2, and you can see there's a lot of growth right here. They're so small, though. I mean, I can't really count these, so it's really hard unless I were to look under a microscope to see if they're supposed to be all individual or if these bigger ones, you know, you can see them here more clearly. There's one there, and then there's one up here. Those are bigger ones, and maybe they could be something else inside. I don't know. I mean, they're the same color, they're just different size, so I don't know. This one, you'll see it too. There's one there at the top, and then I think there's another one in there, like about right here. I don't know if you can see that one. It's smaller. There it is, right there. So that's 10-4. And we're on to 10-5, which is very, very little. It's really hard to see. There's a couple of those dots again, though. There's one right there, and then there's one up here. See it against my glove? Okay. And then here's 10-6. Right there, there's one of those dots. So I don't know, that could be something different that got in there, because there was mention of possibly getting a different type of microbe or bacteria inside the plates when doing the experiment. So anyway, I'm going to be doing some calculations, and I'm going to use what I have here as a guide to do to making the decision on the colony number. I'm not sure based on the picture if I'll be looking at the right thing, but I'm going to do my best. Thank you.